All right, let's get into it. You know, when we think about insulin, we usually hear about this one big eureka moment back in 1921. But honestly, that's just the first chapter. The real story is this incredible century-long saga of reinvention. I mean, each new breakthrough was basically built to fix the problems of the one before it, and in the process, it completely changed medicine as we know it. And the whole story really kicks off with a modern-day mystery. Just take a look at this. Back in 1982, scientists pulled off something incredible. They created perfect, genetically engineered human insulin. This was the discovery that launched the entire biotech industry. But fast forward to today, and that revolutionary stuff, it's barely even used. It's less than 10% of the market. The other 90+%, that's something totally different. So that's the big question we're going to tackle. If making human insulin in a lab was this massive, game-changing achievement, why on earth did we pretty much immediately turn around and try to replace it? Well, to figure that out, we've got to go all the way back to the start. Okay, to really get why the first version of insulin was such a revolution, you first have to understand just how desperate things were. We're talking before the 1920s. Back then, getting a type 1 diabetes diagnosis, well, it was basically a death sentence. I mean, just look at the difference here. The only treatment doctors had was basically a starvation diet. The whole idea was just to slow down the body's inevitable breakdown. You had patients, a lot of them just kids, literally wasting away to nothing. And then, out of nowhere, comes this crude, messy extract from animal guts that could pull them back from the very edge. It wasn't a cure, not by a long shot, but it was a chance to live. Now, we often hear the names Frederick Banting and Charles Best. And for good reason. They were the ones at the University of Toronto with the initial idea, doing these brutal, grueling experiments on dogs. But here's the thing, they didn't do it alone. John MacLeod gave them the lab space and the expert advice. And honestly, the real unsung hero here was James Collip, a biochemist. He was the one who figured out how to turn this messy, toxic gunk into something pure enough that you could actually inject it into a person safely. That was the game changer. And the results? I mean, they were almost biblical. Think about this. January 1922. There's a 14-year-old boy, Leonard Thompson. He's on the verge of death, weighs just 65 pounds. He gets an injection of Collip's new, purified extract. And just like that, his blood sugar plummets. He starts eating again. He starts gaining weight. He starts living. You really can't overstate how huge this was. It was like watching a resurrection. Okay, so you've got a miracle in a lab. That's amazing. But how do you get it to the rest of the world? That's the next big hurdle, turning this complicated, messy lab experiment into a reliable medicine that could be mass-produced. And here's something that's just incredible, especially when you think about it today. The Toronto team wasn't trying to get rich off this, not at all. They were actually terrified that some company would get a monopoly and jack up the price so nobody could afford it. So Banting, Best, and Collip did something amazing. They sold the patent to the university for $1 each, just $1. They wanted to make sure it was a public trust, available to everyone. So to actually scale this thing up, the university needed a partner with some serious manufacturing power. And they found one in the American company Eli Lilly. This is where another key innovation comes in. Lilly's head chemist, a guy named George Walden, he developed a new technique called isoelectric precipitation. And this was huge because it let them produce way purer, super consistent insulin on an industrial scale, something the little university lab could only dream of. And you can see the results of that partnership right here. Because they got that manufacturing process down so early and so efficiently, Eli Lilly basically owned the insulin market for the next 50 years. I mean, look at this. By 1980, they, along with a couple of Danish companies, Novo and Nordisk, had pretty much the entire global supply on lockdown. And for decades, this whole system just worked, but it had a major weakness. It all depended on one single source, pancreases from cows and pigs. And by the time we get to the 1970s, you could really start to see the cracks forming in that system. The stage was set for the next big reinvention. So here were the two big problems. First, animal insulin isn't exactly the same as human insulin. It's close, but not perfect, and that meant some people had pretty bad allergic reactions to it. And second, the supply was just finite. Think about it. You needed the pancreases from about 30 animals just for one patient for 1 year.
As diabetes became more common, you could see a supply crisis on the horizon.
The solution, when it came, was from a totally new science, genetic engineering. And the key idea was something called recombinant DNA. Here's a simple way to think about it. Imagine DNA is a cookbook. Each gene is a recipe for a protein, right? Like the recipe for insulin. What recombinant DNA lets you do is literally copy the recipe for insulin out of the human cookbook and paste it right into the cookbook of something simple, like a bacteria.
So how did they actually do it? Well, basically scientists used special enzymes, kind of like molecular scissors, to cut open a little ring of DNA from a bacteria. They used the same scissors to snip out the human gene for insulin. Because they used the same tool, the two pieces of DNA had matching sticky ends and would just naturally snap together. You then pop that new hybrid DNA back into a common bacteria like E. coli, and then you just let it do what bacteria do best, multiply. And every time it does, it's following that new recipe, cranking out perfect human insulin. You've essentially turned these tiny bacteria into millions of little insulin factories.
And what's just wild is how fast this all happened. Look at this timeline. The basic science is figured out in 72. A tiny startup called Genentech pops up in 76 to try and make a product out of it. Just 2 years later, they've made human insulin in their lab. And by 1982, after teaming up with the powerhouse Eli Lilly, their drug Humulin gets FDA approval. This wasn't just a new drug. It was the first ever biotech drug. It basically gave birth to an entire industry. Which, of course, brings us all the way back to that question from the beginning. We did it. We solved the supply problem. We got rid of the allergy issue. We now have an unlimited supply of perfect human insulin. So, why did we abandon it? Well, the answer lies in this huge shift in how scientists started to think. Once they figured out they could write and edit these genetic recipes, a light bulb went off. They realized the real power of this technology wasn't just copying nature perfectly, it was improving on nature. This chart really lays out the whole evolution perfectly. The big plus for animal insulin? It existed. That was it. For human insulin, the advantage was being pure and having a limitless supply. But then you get to that last column, insulin analogs. And their secret weapon? They're customizable. So what exactly is an analog? It's basically human insulin, but with a tiny strategic tweak made to its genetic code. By changing just an amino acid or two, scientists could design totally new versions. They could make one that works super fast right after a meal, or they could design another one that works incredibly slowly, providing a steady dose all day long. For the first time, they could really start to mimic how a healthy pancreas actually works, which gives people way better control over their blood sugar. So when you step back and look at this whole 100-year journey, a few big ideas really stand out. First, that initial promise of genetic engineering, it wasn't about making a better drug. It was just about finding a more efficient way to make the same drug. Second, that deal between the tiny startup Genentech and the giant Eli Lilly, that basically wrote the playbook for how biotech and big pharma have worked together ever since. And finally, the real lasting revolution wasn't copying nature. It was the moment we realized we could start designing medicines that are even better. You know, in a way, the story of insulin is really the story of modern medicine itself. It's this constant cycle of solving one problem, which then lets you see the next one you have to tackle. It really leaves us with a huge question for the future, right? We've gone from messy animal extracts to designing drugs atom by atom. So what's the next miracle that this new power is going to unlock?